Transcription, going from DNA to RNA. So transcription can be broken down into 4 basic steps. First, there is the initiation, then elongation, so we're actually building the strand. It's got to get stopped at some point, termination, and then there's some final processing that is going to have to take place. So this is post-transcriptional modifications. Initiation, the strand that contains the gene, is called the sense strand. So we're going to bounce back and forth between the different names for these 2 strands. You got double strand. Each one has different names depending on the context you're using. You could be using one name or the other. So the strand that contains the gene is called the sense strand. This is a strand that mRNA ends up looking like. So if the mRNA is going to look like it, then it has to be built, the mRNA has to be built from the other strand. That strand can be called the antisense strand. And again, its pattern itself doesn't code for something, generally speaking. It is trying to be used to make, it's like a reflection of the sense strand. And so therefore, if you take a reflection of the reflection, you get back to your sense strand. So RNA polymerase is the machine that is going to be building this RNA, and we don't need any primers with this one because we're not trying to make DNA, we're just making RNA, so that's all we need to do is use RNA polymerase, start building RNA. That's the goal and the end game. So it attaches the DNA at a special binding site, so there's these promoters that are part of the DNA that are not actually part of the gene. So they're kind of like a little signal that says, okay, start here. And that is going to work for RNA polymerase to essentially land. This is also a mechanism of control as to, we don't want all of our genes being expressed all of the time. We've got genes for everything, right? And so we don't want to make the proteins for all of that stuff at the same time. We want to make it for when it is needed. And so these promoter sequences, as well as other things like them, are able to control which genes are going to be expressed when. So these tend to be high in thymine and adenine, and so we have these what are called TATA boxes, TATA, and this tends to function as a promoter region, a landing point for RNA polymerase. This promoter helps to orient RNA polymerase in the right strand. So again, looking for that, again, we want to be copying the antisense strand, making a copy of the antisense strand so that the copy looks like the sense strand. So if you had your DNA, and again, this is the double helix portion right here. There's 2 strands to it. And as it uncoils, we're able to actually make the mRNA from it. So the one on the right, drawn from this particular picture, this is the antisense strand. And so what that means is the sequence on the left, that's the one we want our mRNA to end up looking like, which means we have to copy the mirror version of it, the complementary strand to it. And so the antisense strand is the one, it's the non-coding one is what it's referred to as, but probably the best name that I would use for it would be called the template strand. It's the one you're going to be using as a template to make the mRNA. The other one, also known as the sense strand, can be referred to as the coding strand, the non-template strand. This is the one that the mRNA is going to end up looking like. And so for common names, sense strand is probably a pretty good name to go with for the one that is not being copied, but is going to have the pattern of nucleotides, minus the thymine uracil change. It'll have the pattern of nucleotides that the mRNA will end up carrying. And that's because the mRNA is looking at the antisense strand, and it's using it as a template to get that sequence. So in the double helix, you got these double helix, so 2 strands. It's not that one of them always has the sense strand and one of them is always the template strand. It's going to vary back and forth between the 2, depending on where the genes are. The genes are located throughout that run of strands. And so, for example, this one here, we have the template strand happens to be the bottom strand for this gene 1, and for some other gene down the line, gene 2, is the other side that happens to be the template strand. So it's not that one single side of the double helix is the sense and one is the template. It is that it varies back and forth, so you have to look at it for each particular gene. And again, the one that the mRNA is using as a template, that's the template strand. So with transcription, we have this particular part that has to be transcribed. That's going to be the gene. And again, at the beginning, there's going to be this promoter. That is where RNA polymerase is going to land. And so it's going to land on the promoter. And again, this tells RNA polymerase where to begin the copying, to begin initiation. Once it is bound, so again, it binds on the promoter, it is going to open up the DNA and begin placing those RNA nucleotides to build the mRNA strand. And as a polymerase, like the other ones, it's adding to the 3' end. So it has to start, in this case here, if it is going to be moving in this direction, if this is the gene that needs to be copied, and over here is the promoter, where the RNA polymerase is going to land, this sort of globby guy here. This is the 3' end of the bottom strand, and so this is the 5' end of the, there's my pen, there it is, 5' end of this first little bit of RNA, meaning this is the 3' end, meaning that the RNA can be built in this direction as well. So like with other polymerases, it's adding onto the 3' end. So it'll move this way and copy the entire segment, the entire gene, reading the template strand, ending up with an mRNA that looks like the coding strand. Elongation, this is where it's actually going about adding on those nucleotides. So RNA polymerase keeps adding them, and again, it is moving from the 5' to the 3' direction because the template strand is running anti-parallel to that, and so as it matches up, so if there was a A here, it'd be matching up a U there. If there's a G here, it acts up a C there, and it is moving in that direction. It is trying to copy this particular gene, and therefore if it is going to be doing that, because it builds in the 5' to 3', it has to be using the bottom strand in order to get that done. So RNA polymerase moves along. New RNA strands are going to be hanging off, and this can happen multiple times. So many RNA polymerases can do this. So as one goes through, another one could come and land here, and then it could start, and you could end up with a sort of train of RNA polymerases going through, making a whole bunch of mRNAs that have the same message in them. If any of them are wrong, it's not a big deal because, again, you can just make a new copy of it. We're not talking about an error in the original DNA. That's a problem. But an error in mRNA is no big deal. So termination, once the RNA polymerase has made its way across the gene, the sequence to be copied into mRNA or transcribed into mRNA, it's going to reach a terminator sequence at the end of the gene, which is going to cause it to disengage from it. It's kind of like the opposite of the promoter region. Promoter brings it in, the terminator sequence sends it away. At this point, it's released, but it will be back, and RNA polymerase is able to start again, maybe somewhere else or maybe on the same gene, and copy another strand of mRNA. Processing is what happens to the mRNA as it is before it is being used to make the protein, and so at that point, just after it's been copied, it's called pre-mRNA, before the processing happens. In order to get it ready to be in the cytoplasm, it's going to need a 5' cap, which is a bunch of G nucleotides or something similar to G nucleotides. It is going to need a poly A tail on the 3' end. This keeps it from getting degraded in the cytoplasm. And these protect it from the enzymes because, again, our cells don't like random RNA being around like what a virus might put in there, and so we try to label it and protect it so that we can recognize it as our own and make it into proteins that we do want. Splicing can also happen, so we have these spliceosomes that are actually made up of another type of RNA, many types of RNA, and they work with proteins, and they will take out, because the pre-mRNA still has the introns and the exons, so they'll take out those introns, and again, it can vary where this is going to be happening, but it will essentially chop it up so that the exons themselves are what remain, and that's what's going to be used to make the polypeptide. Cuts out introns, some exons, and again, you can have a lower number of genes producing a higher number of proteins, as we do with humans, because a gene can be used more than once depending on how you treat the introns and the exons. If for some of them you take out, let's say take out all the introns, you'd get one particular version of the mRNA for the protein, but maybe if you left some of them in, you'd have the same original information, but you'd be leaving some of those introns in, and so you'd have a different version of the mRNA, which could make a different protein. So we have fewer genes, they can actually make more proteins based on how the mRNA is processed. Even if it's the same mRNA piece, it could be processed differently, getting a different protein. Finished mRNA is then moved out of the nucleus into the cytoplasm, where translation can happen. So summary of the process here, start with your DNA. It is going to be transcribed into the mRNA. And again, we are using the template strand to do that. So this is our template strand that's being used to make the mRNA. And the mRNA will look like, and look at it, the A and the U and the T and the G and the G, the A, the A. The mRNA looks like the sense strand of the DNA. This is then taken out and processed. 5' cap, poly A tail, splicing, and exported into the cytoplasm to be picked up by the ribosomes to be read and made and used to make protein.